Hi everyone, my name is Lauren Holian and I am a PhD student in Dr. Tad Dallas's lab at the University of South Carolina. This is my poster presentation from the ESA conference of 2021. In this study, we investigated the interaction of spatial and temporal patterns in abundance-occupancy relationships. Abundance-occupancy relationships predict that species' regional distributions are positively linked to their local abundances. So species with a relatively broad distribution would be expected to have a relatively high local abundance. While these relationships have been described as a common macroecological pattern, the temporal processes that may impact these relationships have received less attention. So here we use NEON's small mammal dataset to explore how temporal occupancy or species' ability to persist on a landscape may complement that more traditional presence-absence data. Figure 1, Panel A, illustrates how we approach spatial and temporal occupancy in this study. Here, spatial occupancy was quantified as the fraction of sites a species occupies. So for this example, all sites would be occupied. And for temporal occupancy, we looked within sites. Specifically, we quantified the fraction of months a species was present in a site in each given year. And this is represented in this figure as the multiple time slices associated with a site. To achieve comparable landscape values, though, we took the mean across sites for both spatial and temporal occupancy. And then Panel B conceptualizes how this might result in potential life histories given the differences in spatial distribution and temporal persistence. So this could range from a spatially limited and temporary species on the bottom left to a persistent generalist in the upper right. And then we extended this to predict that these combinations would correspond to varying local abundances. We characterized the abundance-occupancy relationships of 53 small mammal species from 2014 to 2019. We did so through linear regressions and assessed the relationships both intra- and inter-specifically. Intraspecific regressions are single-species relationships where the individual values represent different years, and interspecific regressions are relationships across multiple species within a single year. So figure 2 shows our intra and interspecific relationships that we observed. Intraspecific regressions are indicated in the pink for positive relationships or blue for a negative relationship. Interspecific regressions correspond to the yellow lines alone. All interspecific relationships were positive and significant, with the exception of the 2019 spatial regression. Intraspecific relationships, on the other hand, were less clear, and we observed both positive and negative relationships. Many of these, however, were not significant due to data limitations. Figure 3 is a plot of the temporal and spatial occupancy of all species across all years. Each circle represents a species for a given year, and the circle size indicates the relative local abundance of that species in that year. You can see here how the highest abundances tend to occur either when spatial or temporal occupancy, or both, are also high. Temporal occupancy also seems to be more evenly distributed, while spatial occupancy was more skewed, with few species occupying more than 40% of sampled sites. Dipodomus mariami, a desert rodent, is a great example of how temporal processes may be particularly important for understanding local abundance. This species is spatially restricted, only occupying about 4% of the sampled sites, but it achieved one of the highest abundances. So, the main things to take away from this work are how both spatial and temporal occupancy were positively related to local abundance across small mammal species. Though this was less clear for the intraspecific relationships, we saw really consistent positive relationships in the interspecific relationships across different years. Spatial and temporal occupancy exhibited different distributions across species. In the case of high abundance and low spatial occupancy, such as that was pointed out by Dipodomus mariami, those instances suggest that temporal occupancy is of particular significance when trying to understand patterns of abundance. This work could not have been accomplished without the assistance of Dr. Tad Dallas and Kleber Tenkatan and funding from the NSF. Thank you all for listening.